May 1993, Four Corners Region, New Mexico, USA. A 19-year-old Navajo athlete named Merrill Behe is driving to the funeral of his fiancée. She died 5 days ago. She was young, healthy, athletic, no underlying conditions, no warning signs. She woke up with what felt like the flu. Fever, muscle aches, fatigue. Within 48 hours she could not breathe. Her lungs were filling with fluid so rapidly that her oxygen levels collapsed. She died before they understood what was killing her. Now Merrill, grieving but physically fine, is on his way to her funeral. And he feels it. The same feeling. The ache behind the eyes. The heaviness in the chest. The fever coming on fast. He collapses. An ambulance takes him to the same hospital. The same doctors are on duty. They see the same thing. His lungs filling, oxygen dropping, blood pressure crashing. They put him on a ventilator. It does not help. Within hours, Merrill Behe is dead. Both of them, young, healthy, athletic, killed within days by something that destroyed their lungs. The doctors knew they were looking at something they had never seen before. Something with no name. Something that killed fast, killed young, killed healthy people. Something that was already out there, in the air, in the dust, in the invisible spaces of the American Southwest, waiting. They had no idea how many more it found. To understand hantavirus, what it does, where it comes from, why it terrifies scientists, you need to understand the relationship between viruses and animals. Hantavirus is not new. It didn't emerge from a lab or mutate suddenly. It has lived inside rodents for millions of years, tens of millions of years. The virus and rodent evolved together, side by side, over geological time. A perfectly balanced relationship where the rodent does not get sick. The rodent carries the virus in its saliva, urine, feces, and feels nothing. Its immune system and the virus reached an evolutionary truce. The catastrophic problem is what happens when that virus leaves the rodent, when it encounters a species that has never met it before. A species like us. When hantavirus enters a human body, it doesn't settle quietly. It triggers a violent, overblown response. The immune system destroys the lungs. The medical term is a cytokine storm, an immune response so intense it causes more damage than the virus itself. Lungs fill with fluid. Blood vessels leak. Oxygen stops reaching the blood. A healthy person, an athlete, a farmer, a hiker, a child, can go from fine to dead in less than 72 hours. There is no vaccine, no specific antiviral treatment, only supportive care, ventilators, oxygen, fluid management, and hope. Even with the best medical care, the mortality rate of the most dangerous hantavirus disease is between 35 and 40%. You read that correctly. Between 35 and 40% of people who develop full hantavirus pulmonary syndrome die. This virus kills more than 1 in 3 of the people it infects. That number should terrify you. And here's what should terrify you even more. It is almost certainly spreading in ways we are not fully tracking. The 1993 outbreak was not the first time hantavirus killed humans. It was just the first time American doctors recognized what they were seeing. Hantavirus had been killing people for a very long time, in ways medicine had not connected. The Korean War, 1950 to 1953. American and UN soldiers began dying of a mysterious hemorrhagic fever that caused kidney failure, massive internal bleeding, and death. Thousands of soldiers were affected. The disease was called Korean hemorrhagic fever, and nobody understood what caused it. A Soviet scientist, Mikhail Chumakov, suspected a rodent-borne virus as early as the 1950s. But Cold War politics meant scientists weren't sharing data. 40 years of potential research lost to geopolitics. When American virologist Dr. Ho Wang Li isolated the virus in 1976, he named it Hantavirus, after the Hantan River in South Korea, near where the first cases were identified. The family of viruses became known as hantaviruses. Even after identification, the assumption was it was primarily an Old World problem, a Korean problem, an Asian problem. When cases appeared in New Mexico in 1993, young Navajo people dying of a lung disease, it looked nothing like the kidney disease in Asia. The initial response of American medicine was confusion. Nobody was looking for hantavirus in the American Southwest. Nobody had told them to look. In that gap between what science knew and what medicine was prepared for, people died. The investigation that followed was extraordinary. A team of CDC investigators descended on the Four Corners region, trying to understand what was killing these young, healthy Navajo people. They tested blood samples, interviewed survivors and families, trapped rodents, tested the rodents. Within weeks, they identified the culprit, a previously unknown species of hantavirus, carried by a small brown mouse called the deer mouse, Paramyscus maniculatus, one of the most common rodents in North America, found in every state except the southeastern coastal plain. It lives in fields, forests, deserts, mountains. It lives in barns, woodpiles, walls of houses, gaps under floorboards. It is everywhere. A significant percentage of deer mouse populations carry this virus. The new virus was named Sin Nombre virus, Spanish for virus with no name. The disease it caused was hantavirus pulmonary syndrome, HPS. In the initial 1993 outbreak, 24 people were infected. 13 died, a mortality rate of 54%, more than half. In the months and years that followed, doctors across the country began looking for cases they had missed. Hantavirus had been killing Americans quietly for years. Cases were found in 34 states. Every single one linked to contact with rodent droppings, urine, or nesting material. Everyone preventable. With knowledge nobody had given the public, because nobody had known to give it. Now, this story moves from history to urgent present. What happened in 1993 was not an isolated event. It was a preview, a warning, a demonstration of the threat infectious disease specialists warned about. For 30 years, governments have consistently, catastrophically underfunded and underprepared. Since 1993, hantavirus has continued to spread and evolve. New variants have been identified on every inhabited continent. In South America, particularly Argentina, Chile, Brazil, and Panama, Andes virus emerged, and Andes virus has a characteristic Sin Nombre virus does not have, a characteristic that sent a chill through the virology community. Andes virus can spread from person to person, not efficiently, not easily, not like COVID-19 or influenza, but it can do it. There are documented cases, confirmed, peer-reviewed, published in major medical journals, of Andes virus transmission between people, between patients and healthcare workers, between family members who cared for a sick relative, between sexual partners. The virus that was supposed to only spread from rodents to humans has, in one strain, learned to move between humans. It has not yet learned to do it efficiently. The key word is yet. Viruses do not stay the same. Viruses mutate. Viruses adapt. Viruses find new pathways when old ones are blocked, and exploit new opportunities. The question virologists ask with increasing urgency: what if Sin Nombre or other lethal strains acquire human-to-human spread? What happens if a virus that kills 35 to 40% of people it infects develops efficient human-to-human transmission? The answer is why this documentary exists. The answer is why scientists describe it as one of the most dangerous pathogens on Earth. And the answer, with 2024 data, is what Parts 2 and 3 are about. What changed in 2024 and why scientists are more worried about hantavirus now than since 1993 comes down to 3 converging factors. Individually concerning, but together creating a picture that demands attention. The first factor is climate change. The relationship between hantavirus and climate is direct, documented, and accelerating. Hantavirus spreads when rodent populations explode. Rodent populations explode when food is abundant. Food becomes abundant when certain climate conditions produce large quantities of seeds, nuts, and vegetation. In the American Southwest, El Niño produces wet winters and springs, leading to abundant vegetation, abundant food for mice, and population explosions, leading to increased human exposure to infected rodents and their droppings. Climate change is making El Niño events more frequent and intense, which means rodent population explosions in hantavirus endemic regions are becoming more frequent and intense. The CDC's own modeling data projects increased risk through 2050 as climate patterns shift. The second factor is habitat encroachment. Human populations expand into previously wild areas. Suburban development pushes into forest edges and desert margins. Outdoor recreation increases in hantavirus endemic regions. The interface between humans and infected rodent populations is growing. More people in more places where infected mice live means more opportunities for exposure. And the third factor, the one that keeps virologists awake at night, is the mutation question. In 2024, genomic sequencing tracks viral evolution in real time, at a resolution impossible even 10 years ago. What that sequencing is showing in hantavirus strains from South America, Asia, Europe, is continuous mutation. The virus is not standing still. It is searching. Whether it will find the specific combination of mutations for efficient human-to-human transmission, nobody can say. But the scientists watching it most closely are not reassured. Part 2 will tell you exactly what they are seeing, and why 2024 may be the year the conversation about hantavirus finally becomes the conversation it should have been 30 years ago. March 2024. Yunnan Province, China. A research team from the Wuhan Institute of Virology publishes a paper in a peer-reviewed scientific journal. The paper describes the discovery of 24 previously unknown strains of hantavirus, identified through genomic sequencing of rodent populations across southern China. 24 new strains, in one study, in one region, of one country. The paper notes that several of these new strains show genetic characteristics that are concerning. In the world of virology, where understatement is a professional habit, concerning, from a team of researchers studying novel rodent-borne viruses, means something. It means they saw something in those genetic sequences that made them pause. That made them look again. That made them write it down and submit it for peer review. The paper received almost no mainstream media coverage. It was not reported by CNN or the New York Times or the BBC. 24 new strains of one of the deadliest viruses on Earth, discovered in the same region where COVID-19 emerged, and the world did not notice. Because the world was not looking, because nobody told them to look, and because the systems that are supposed to translate scientific alarm into public awareness failed. Let me tell you what is actually happening with hantavirus around the world right now, in 2024. In Argentina, case numbers in 2023 and early 2024 were the highest recorded since surveillance began. The El Niño event of 2023 to 2024 drove rodent population explosions across the Pampas and Patagonian regions, producing a surge in human cases that overwhelmed local health systems in several provinces. The Argentine Ministry of Health issued emergency advisories. They documented a cluster of cases in the Patagonia region that epidemiologists believe may include instances of person-to-person transmission. Not confirmed, under investigation, but enough of a signal that the researchers tracking it are watching it very closely. In Chile, Argentina's neighbor, which shares the Andes mountain ecosystem where the virus is most prevalent, the situation is similar. Case numbers up, rodent populations up, and a healthcare system exhausted by COVID-19, now facing a resurgent hantavirus threat with depleted resources. In Panama, where a strain called choclovirus circulates, surveillance data from 2023 shows increasing case numbers in the Azuero Peninsula region, linked to agricultural workers with rodent exposure. Panama is significant because of its geography, a narrow land bridge connecting North and South America. What circulates in Panama does not stay in Panama. In the United States, the 2024 picture is concerning in ways not reflected in headline case numbers. The CDC reports approximately 40 to 50 confirmed cases of hantavirus pulmonary syndrome per year, with a mortality rate around 35%. Those numbers seem small. They are not small. A disease that kills more than 1 in 3 of the people it seriously infects, with no vaccine and no specific treatment. But beyond the headline numbers, there is a surveillance gap that infectious disease specialists have been flagging for years. Hantavirus pulmonary syndrome is notoriously difficult to diagnose in its early stages. The initial symptoms are indistinguishable from influenza or COVID-19. A patient presenting with early HPS in an emergency room in rural New Mexico or Montana is almost certainly going to be sent home with a diagnosis of flu, and may be dead before anyone realizes what was actually killing them. Epidemiologists believe the official case count significantly undercounts the true number of infections. The real number of Americans who die from hantavirus every year may be higher than the official statistics show, and nobody knows by how much, because nobody is systematically looking. Now, I need to tell you about the research that is being done, and, more importantly, about the research that is not being done. The gap between what science knows about hantavirus and what governments are funding to address it is one of the most striking examples of pandemic preparedness failure. The research community studying hantavirus is small, extraordinarily small for a pathogen of this danger level. The funding available for hantavirus research is a fraction of what is available for higher-profile pathogens. After COVID-19 temporarily increased awareness, there was a brief surge of interest in hantavirus funding. That surge has already dissipated. The political will to maintain elevated attention to emerging infectious disease threats has eroded, and the scientists who study hantavirus are watching the funding contracts, warning that the window of post-COVID attention that could have been used to make real progress on a hantavirus vaccine or treatment is closing. Dr. Brian Hjella said in a 2023 interview that the scientific community's understanding of hantavirus has advanced enormously since 1993, but that the translation of that understanding into actual medical countermeasures, vaccines, antivirals, diagnostic tools, has lagged decades behind. He said the virus is better understood and less prepared for than it has ever been. Better understood and less prepared for. Hold that phrase, because it describes not just hantavirus but the entire architecture of global pandemic preparedness in 2024. A system that generates knowledge faster than it generates action, that understands threats more clearly than it addresses them. That learned the lessons of COVID-19 in its scientific literature but has not yet learned them in its budgets, its infrastructure, or its political will. The genomic surveillance data coming out of 2023 and 2024 is the thing that is generating the most concern within the hantavirus research community right now, because genomic sequencing has revealed something about the rate and direction of hantavirus evolution that was not fully visible before. Hantaviruses are RNA viruses. RNA viruses mutate faster than DNA viruses because the enzyme they use to copy their genetic material makes more errors. More errors per replication means more mutations per generation, means faster evolution. HIV is an RNA virus. Influenza is an RNA virus. SARS-CoV-2 is an RNA virus. Hantavirus is an RNA virus. And the sequencing data from 2023 and 2024 is showing that hantavirus strains are evolving at rates faster than predicted. The virus is not just mutating. It is exploring. In the language of evolutionary biology, It is searching through genetic space, trying combinations, discarding what does not work, retaining what does. Most mutations are neutral or harmful to the virus, but some are not. Some are improvements. Some allow the virus to infect new cell types, to evade immune responses, to survive longer in the environment, to replicate more efficiently. And some, the mutations that keep infectious disease specialists awake at night, some improve transmissibility. The ability to spread from one host to another, the ability to move from a mouse to a human with less contact, the ability to move from a human to another human with more efficiency. Nobody is saying that hantavirus has acquired efficient human-to-human transmission. It has not. The current data does not show that. What the current data shows is a virus that is actively evolving, in multiple strains simultaneously, in a direction that warrants urgent, sustained, well-funded scientific attention. And what the current reality shows is a world that is not providing that attention, not at the scale the threat demands, not even close. Let me tell you about one more thing that happened in 2024 that did not make the news, but that people who track emerging infectious disease threats found deeply unsettling. In February 2024, a cluster of hantavirus cases was identified in a rural community in the central highlands of Bolivia. The cluster involved 7 people, all from the same small farming community, all with documented rodent exposure from working in agricultural storage facilities. 5 of them developed hantavirus pulmonary syndrome. 3 of them died. The mortality rate in that cluster, 60%, was higher than typical. Bolivian health authorities and the Pan American Health Organization investigated. They collected samples. They sent them for genomic sequencing. The sequencing results showed that the strain responsible for the Bolivian cluster had several genetic features not seen in previously characterized South American hantavirus strains. Whether those features are responsible for the higher mortality rate, whether they represent a meaningful evolutionary development or a statistical anomaly, these questions are under investigation. The investigation is ongoing. The results have not been published. The public has not been informed. And this is not a conspiracy. This is not a cover-up. This is simply how the early stages of emerging infectious disease surveillance work. Cautiously, methodically, in the peer-reviewed literature rather than in press releases. But it is also a reminder, a stark, uncomfortable reminder, that somewhere right now, in a farming community or a forest or a laboratory or a city market, the next pandemic pathogen is already circulating, already mutating, already finding its way, and the question is not whether we will face another pandemic. The question, the only question that matters, is whether we will be ready when it comes. Part 3 has the answer, and it is not the answer you are hoping for. Let me ask you a question. Not a rhetorical question. A real question I want you to sit with for a moment before we continue. In January 2020, if someone had told you a novel coronavirus would kill millions, collapse the economy, and change the world, would you have believed them? Most would not have believed it. Yet the science was there. Pandemic preparedness reports and modeling were all available. Bill Gates warned in 2015 that the world was unprepared for a respiratory pandemic. He was right. The warnings were right, but the world was not ready. Now, I'll share what hantavirus scientists say privately about its risk to humanity in the next decade. These conversations don't make the news. Apply the same standard to their words as you would to pre-COVID warnings. Hantavirus warnings are not new. They've existed for 30 years, since 1993. Since a 19-year-old Navajo athlete died in New Mexico. The warnings have been there, but the world has not been listening. Here's the scenario infectious disease experts describe: a hantavirus strain acquiring efficient human-to-human transmission. Not a certainty, but a grounded what if. The scenario begins with a mutation event. In rodent populations across continents, a hantavirus strain gradually mutates. improving its human spread. The first sign is a cluster, like those in Bolivia, Argentina, and Chile, where transmission patterns don't fit rodent-to-human exposure. Family clusters grow, healthcare workers fall ill. The cluster is investigated. Samples are collected, sequencing is done. In the genetic sequences, something new appears, recognized by scientists as significant. This is not hypothetical. It's almost exactly what happened with COVID-19. A Wuhan cluster, unexpected cases, samples, sequences, and a novel virus capable of efficient human transmission. The difference between a manageable outbreak and global catastrophe depends on the response, how fast the alarm is raised, how seriously it's taken, and how quickly governments act. How much infrastructure, diagnostics, treatments, vaccines, supply chains, communication is in place before the alarm. For COVID-19, answers were devastating. For hantavirus in 2024, they are worse. Here's why it's worse. COVID-19 spreads through air via droplets and aerosols, the most efficient route. It spread globally within weeks. The global public health infrastructure had extensive experience with respiratory pathogens, with frameworks and protocols from SARS, H1N1, MERS, and influenza. Hantavirus doesn't spread like COVID-19. It spreads primarily through contact with rodent excreta, aerosolized when disturbed in enclosed spaces. This transmission route has less developed surveillance, less trained clinical recognition, and less rehearsed public health response protocols. When hantavirus acquires human-to-human transmission, early cases will resemble severe flu or pneumonia, which is exactly what hantavirus pulmonary syndrome looks like. In a world exhausted by COVID-19, with eroded trust and weakened political will, early hantavirus cases will almost certainly be misidentified, misattributed, and missed. By the time the pattern is recognized, the virus will have had days or weeks of undetected spread. For a pathogen with a 35 to 40% mortality rate, this means catastrophe. Precision matters. The scientific consensus in 2024 is that hantavirus doesn't currently pose an imminent pandemic threat. Current strains lack efficient human-to-human transmission. The required mutations are specific and not yet observed. The probability is low, but significantly higher than 0 and higher than 10 years ago. However, the consequences of being wrong are catastrophic. The cost of preparedness is modest compared to the consequences, and current preparedness is inadequate. The rational response is to dramatically increase investment in hantavirus research, surveillance, diagnostics, and vaccine development immediately. Not because a pandemic is certain, but because the downside risk of inadequate preparation, as COVID-19 showed, is millions of lives and trillions of dollars. The upside of preparation, vaccines, treatments, surveillance systems, strengthens readiness for the next pathogen, whatever it may be. This benefit is incalculable. Scientists say prepare, not run, but governments are not preparing adequately, not urgently, not in proportion to the risk. Here's the vaccine situation in 2024. A story of extraordinary scientific progress happening in obscurity, deserving to be told. No approved hantavirus vaccine exists for human use globally. 2 inactivated vaccines in China and South Korea protect against the Asian strain, but with limited efficacy. They require multiple doses and don't protect against American or South American strains, which kill 35 to 40% of seriously infected Americans. For these, there is no vaccine. However, mRNA vaccine technology, developed for COVID-19, has transformed hantavirus vaccine development, offering cautious hope. Before COVID-19, vaccine development took 10-15 years. The mRNA platform can produce vaccine candidates in weeks. Scientists design mRNA sequences to instruct human cells to produce a piece of the pathogen. which the immune system learns to fight. In 2022-2023, research teams produced mRNA vaccine candidates for Nombre and Andes viruses. Animal studies showed promising results. Human clinical trials haven't begun. Funding is the limiting factor, not science. The science is there, the platform proven, candidates exist. What's missing is political will and financial commitment to move candidates through trials and into production before the crisis, not after. After is too late. The honest, evidence-based prediction from researchers is this. In the next 10-20 years, a significant hantavirus outbreak is more probable than ever. Factors driving this include climate change expanding rodent ranges, global travel spreading novel strains, and ongoing viral evolution. The global pandemic preparedness infrastructure, weakened by fatigue, budget cuts, and political conflict, is less ready than it should be after COVID-19. The most dangerous moment for any infectious disease is not the outbreak, but the calm between outbreaks, when urgency fades and funding dries up. Scientists find themselves speaking to empty rooms. That is exactly where the world is with hantavirus now. The calm between what came before and what may be coming next. I want to tell you about Dr. Christina Spiropoulou, chief of the Viral Special Pathogens branch at the CDC in Atlanta. She studies hemorrhagic fever viruses like Ebola, Marburg, and Hantavirus, and is a leading expert on hantavirus pathogenesis. In a 2023 scientific interview, she was asked what keeps her up at night. She didn't hesitate. Hantavirus. She said hantavirus has the widest gap between its capability and medicine's response. Its 35 to 40% mortality rate demands more attention. High lethality, no vaccine, no specific treatment, and a ubiquitous animal reservoir create a unique threat profile if it acquires human-to-human transmission. She described a virus that kills over 1 in 3 infected, has no vaccine or treatment, lives in rodents globally, is evolving, and has shown human spread in 1 strain. And it exists in a world less ready for it today than it should be after everything COVID-19 taught us.